What does the microbial means? Microbial means constitute microbial organisms. Organisms that are visible only, you can observe the only under electron microscope only. Where you can, what type of that is there? In case you can take the bacteria. In that bacteria, two types are there. One is the gram positive, one is the gram negative, gram negative bacteria. Another one, fungi. The second one is the fungi. That fungi, molds, yeast, mushrooms, protozoas, etc. These are the organisms. What is the biotechnology? Here, okay, organisms are there, all things are the microorganisms. But what is the fungal biotechnology? In the fungi, what is the use of in the biotechnology? How to use in the industry level? In the, many importance are there. Penicillium is there. Penicillium in industry used for this one. That is the main important, that is antibiotic. Penicillium fusaricone used the fungi. This is the importance of the penicillium. And yeast, yeast also the best example for the industrial use. Yeast, cheese product, milk products, to utilizing these are the products used for the fungal industry. And economically very important to the fungus. Why in the sometime food material, mushrooms is the food material, protein cakes, next one fermentation foods, biological assays also, soil, that one. Some are the harmful fungi. Some are the harmful fungi. So these some harmful fungi, very deterioration of particles, staining wood, wood decay, many causing the diseases. These are the harmful fungi. Okay. Now, microbes utilized in this process. What is the microbes? First one bacteria one, second one is fungi. In the bacteria, that are the foregoing consists account categorize different activities of fungi. Bacteria two types. Fungi, our friends and foes. I would like to elaborate the ways in the which fungi are industry useful to us and how these are used in biotechnology. Now, fungal biotechnology. Although the term biotechnology is of recent origin, some biotechnological process and the scientific disciplines which contribute to biotechnology have a longer history, have a very longer history. This biotechnology has been defined by the scientist name Spinks, Spinks, Spinks in 1980 as a application of biological organism systems, biological organism systems. Next one, the biological organism systems are process to manufacturing industry. I'll explain to the what is biotechnology. Now I will explain that one. Okay. In this one, in this process, biotechnological process, manufacturing industry, several other definitions have been attempted. However, irrespective of which definition is used, there can be little doubt that fungi represent an important group of organisms in biotechnology and that mycologists have a contribution to make of biotechnology. It is not unreasonable to suggest that the origin of biotechnology can be identified in some of the traditional industrial process using fungi. The technology developed new products through the manipulation and control total enzyme activities of microbes. This is called genome editing technology, genetic engineering technology, genome technology, CSIPR, case 9, genetic engineering, calling type of this one. Here, in the biotechnology, irrespective of which define the very doubtful. And it is not unreasonable to suggest that the origin of biotechnology can be identified in some of traditional industrial process using traditional industrial process using this one is the using fungi, the use of the yeast. The second one is the yeast product. Use of the yeast, one second, this one. Uh, the use of the uses of the yeast to produce alcoholic beverages, where you can use the produce the yeast. This fungi, diverse group of eukaryotic organism. You know that one. All fungi is the eukaryotic organism that includes yeast, mold, and mushrooms. Yeast, mold, and mushrooms in uh, in uh, in in inhabited various environment ranging from soil, water, living and to living organism, and play crucial roles in nutrient cycling and decomposition. Here, in industry level, in industry, how to use the fungal, the industrial process fungi use of yeast to produce alcoholic beverages. You know, many alcoholic beverages are there, beer, like alcohols, like that. Penicillium to produce the blue cheeses, the cultivation of mushrooms, and the use of the cozy process to produce the soya sauce and other food products have been practiced a commercial process for hundreds, if not thousands of years, and are still carried out in a highly valued industries. Okay. However, modern biotechnology has had to await the development of genetics, biochemistry, and fermentation technology. The production of citric acid by citric acid, you know, vinegar. In that production, citric acid, vinegar or citric acid production by Penicillium and Aspergillus niger. Both are the Ascomycotina member. Penicillium and second one is the Aspergillus niger, represent one of the earliest process exploiting a microorganism to produce the chemical product in aerobic condition. It produced aerobic condition. Uh, one second. Hello. సార్, నేను కుమార్ సార్ క్లాస్లో ఉన్నాను సార్. మీరు నాకు ఇంకా నేను మన ఇద్దరం డిస్కస్ చేయలే కదా అని కుమార్ సార్ది తీసుకుంటలేండి అని చెప్పాను. అదే, మళ్ళీ మీకు ఎప్పుడు ఉంది సార్? 1:30కి తీసుకోనా? రెండు తీసుకోలేకుండా అవుతాను కదా ఇప్పుడు. ఆల్రెడీ దీంట్లో జాయిన్ అయినా. కుమార్ సార్ ఫ్రీగా ఉన్నారు, ఒకసారి కాల్ చేసి తీసుకోమనండి. ఉమ్, అయ్యయ్యో అవునా? ఓ, ఏం లేదు ఒకసారి. అయ్యో, మీరు ఒక్కసారి నైట్ నాతో డిస్కస్ చేసింటే అయిపోయేది, ఇంకా నేను మళ్ళీ డిస్కస్ చేయలే కదా అన్నట్లు నేను ఊరుకున్నాను. కుమార్ సార్ని అడిగిన. మీరు ఒక్క పని చేయండి, ఏం పర్లేదు, కుమార్ సార్కి చెప్పండి తర్వాత క్లాస్ తీసుకుంటే, ఇప్పుడైతే మీరు తీసుకోండి. సార్ క్లాస్లో ఉన్నందుకు సార్ ఫ్రీగా ఉంటాడు. ఆ, నేను చెప్పనా లేకపోతే? ఏం కాదు, సార్కి చెప్పండి, ఏం పర్లేదు. సరే సరే, ఓకే ఓకే సార్. Sorry for the disturbance. Now we will continue to the biotechnology. Here, penicillium is the most important characteristic features of the industry level. In the industry, in this one, in the industry, the process, how to the process is there, what is the product to form in this one. These are things we will discuss that one. Okay. Next one, fungi are the eukaryotic characters, inhabit various environments ranging from soil, water, living organism. Fungal biotechnology refers to utilization of fungi, include yeast, mold, and mushrooms for various industrial, architecture, agricultural, agricultural, pharmaceutical, and environmental applications. These are all involvement in the fungal biotechnology. This introduction to fungal biotechnology, total the fungal involves harnessing, control, and make use of natural resource. The metabolic capabilities of fungi to produce value products, enzymes, bioactive compounds to perform specific tasks such as bioremediation and biocontrol. What are the evidence in this one? What are the evidence are there? Okay, uh, here, main important, this was first carried out to the surface culture. This one in 1983 was put into commercial production in 1923. However, the use surface culture has many limitations as a method of large scale cultivation of fungi. In this method, the cultivation very large method, okay, very large methods are there in that to produce the large methods. And this method, very large method, and this method was, you can identify the, this second method, main important to produce to the surface, that is the attempt was therefore made of cultures of aerobic fungi in submerged cultures also. The development of steroid tank reactors as a large scale operators for the growth of aerobic microorganism in the submerged culture was stimulated during the Second World War by the need to develop the production capacity for penicillium, another fungal fermentation. The development of this industrial fermentation probably represent the being of modern biotechnology. It could solve many technical problems, particularly the development of techniques to transfer large quantities of oxygen into growth medium. Okay, then until fossils and land fungi date to almost 400 million years ago. Until the later half of the 20th century, fungi were classified into plant kingdom. Okay, what is the evidence? Evidence and origin of fungi. Molecular evidence is there, probably evolved are there. Support the hypothesis the fungi, animals, diverging from the common ancestor that was unicellular Bohr flagella. This evidence before colonization of land by multicellular organisms. Multicellular organism. See, this is diagram represent growth and metabolism, universal phylogenetic and universal phylogenetic tree showing relation between major linear of the three domains of life. One domain is the bacteria, second domain archaebacteria, third domain eukarya. Based on salt subunit rRNA gene sequence analysis. This is the fundamental principle how fungi might be used biotechnology. How fungi is used to the biotechnology. Fundamentals. The starting point is a consideration of the both physiological and biochemical features of fungal growth. The growth and metabolism of fungus are indeed that of any microorganism can be represented diagrammatically simple. Carbon skeleton of molecules. Carbon skeleton of molecules means which compose cell structure such as cell wall, proteins, phospholipids. These are the main required for growth are produced by catabolism of the carbon source in the growth medium of the fungus. Usually, this carbon source is how to carbon source to produce carbohydrates, frequently glucose. This process of catabolism is brought about the metabolic events of glycolysis or citric acid cycle by the formation and interconversion of amino acids and the like. The biochemical process which bring about the synthesis of compounds which go to make up the constitutions of the normal growing cell or hyphae. That is called the primary metabolism. That one is called the primary metabolisms. To be more species, to be more specific, primary metabolism involves an interrelation series of enzymes, mediated reactions which provide biosynthetic intermediates and energy and convert biosynthesis intermediate into essential macromolecules. That is the DNA, RNA, proteins, and polysaccharides. Thus, primary metabolisms provide both carbon skeletons as well as energy for the growth. The process of fungal growth is used biotechnology. We speak of the generation that is called biomass. That meaning is called biomass. The cultivation of mushroom is the classic instance. Other example are baker's yeast. Baker's yeast, and Fusarium graminum. Which one is baker's yeast? Fusarium graminum is the baker's yeast. Why? Primary metabolism is the source of carbon skeleton and energy flow growth. Primary metabolites may be of a very considerable commercial and industrial importance. Here come the relatively low molecular weight compounds, which are components of the metabolic pathways which lead to the macromolecules. What are the metabolic? Ethanols, alcohols is the outstanding example for the fungal principles fungal biotechnology. Ethanol production by a fungus Saccharomyces cerevisiae. Which one fungi to ethanol to produce? Saccharomyces cerevisiae occur under normal condition of growth. In case of other metabolites, however, the metabolism has to be perturbed to stimulate the production of the particular compound in sufficient quantity to make the process commercially valuable. The classic case is stimulation of production of glycerol by yeast. Under those conditions, when the organism would normally be producing ethanol, here acetylate is trapped by the addition of sulfate. In this one second. Sorry for the disturbance. This one main is the recently very growing media in fine volumes. Here, acetaldehyde, acetate. This is the acetate is trapped by sulfate. NAD is reduction of dihydroxyacetone to glycerol under normal condition. Okay, and here this tree showing three relation. One is the bacteria, archaebacteria, eukaryote. See, bacteria, thermosobacteria, thermatose, bacteroids, cyanobacteria, protobacteria, fumicates, chlorifex, aquifers. This connection between the archaebacteria. This is the one type of bacteria, archaebacteria, Karyokata, Nanocata, Cenerocata, Tamuriocata, Eukaryota. This is the connection between bacteria, eubacteria and eukarya. Eukarya main is the animals, amoeba, fungal, red algae, plants, green algae, diatom, ciliates, diflagellate, cenozoans, euglenozoans, dimalphates. Connection, this is a universal phylogenetic tree. Connection in between both three. Yes, based on the small subunits. rRNA gene sequence analysis in this one. Okay. Coming to the fungal ecology and diversity. What is the diversity is there in that one? Growing fungal biodiversity is the different diversities are there. The Fungi grow almost every habitat in imaginable along there. Some types of organic, some types of capable of fungi. Organic matter present and the environmental is not to exams. Okay, not to exams. Next one, the play critical role balance in the ecosystem. Balance with the ecosystem. Plant, fungi, animal status, that is the ecosystems are. Next, they are major decomposition of the nature. Next one, fungi powerful player in global biogeochemistry. Recycling, what are that one? Recycling, mobilizing nitrogen, phosphorus, and other bio elements are present in this one. Coming to the another one second, next one. See, you know, general characters, all fungi general characters already. We will discuss about the heterophyte, absorption, the saprophytic, symbiotic, all you know. Next, coming to the another one. This is the structure of fungi. What is the structure? What is this one? All are the fungal structures. Coming to the another one. See, this is the reproduction. Reproduction also already completed. Okay. Now, this one you can observe that one, the fungal between bacteria and virus. What is the features of all organisms to the fungal to bacteria? See, size, virus, bacteria, fungi. This one cell type, acellular virus. Bacteria prokaryotic, fungi eukaryotic. Size, see, you can observe the size also. 0.02 to 0.03 size you can observe the in the viruses. Bacteria only 0.3 to 0.23 to 2 microns. Fungal 3 to 10. 3 to 10. 3 to 10 microns may be large. That is called, you know, large one already, you know, that one is the mushrooms, edible mushrooms. You can observe very large, not to, you can see the under microscope, no, very useful proteinous plants, proteinous plant body. Okay. Next one, cell type, DNA, RNA, either both, any one is there. In bacteria, two are there. In fungi, also two are there. Nucleic acid replication in the virus only host cell. But here, continuous process in the bacteria. Here, G and S phase. In the cell cycle, gap phase and synthesis phase in the fungi. Replication complex binary fusion in this one, mitosis, meiosis. Next one is the organelle use host. Next one, not membrane bound. In this one also not membrane bound. And ribosomes, none in the virus. Here in the bacteria, 70 years, that is a 30, 50. Fungi, eukaryote, no, that's why 80 years, 60 years, 40 years and 60 years. Cell membrane, envelope, sometimes non-envelope. Here, no sterol mycoplasma. Here, cell, this is the ergosterols also present. Cell wall is absent. Peptidoglycan to present cell wall. And next one, chitin is the cell wall. This is the important of fungi, harmful fungi and beneficial fungi. And potential of fungi, this is the most important characteristic feature of the total fungal biotechnology. Now we will, the discussion of the fungal biotechnology is completely completed. Now we will discuss about to the fungal biodegradations. What are the relation fungal in relation to pollution, fungal in relation to the biodegradation. Fungal biodegradation is the most, what is the pollution is control. Fungal pollution and biodegradable. How to control the pollution to fungal. This one also very most important characters for the fungus. And here environment, how to use the fungal use for the environmental conditions. This environmental, you know that one, some of the harmful fungi are there, some of the useful fungus are there. What are the harmful fungus? What is the useful fungus is the most important. This one, fungal is the advanced in chemical, medicinal, and emergentic of the that is the many fungal biodegradation agents. Many are there. Now I will share the my PPT. One second. Fungal degradation method, biodegradation. I will explain. Share my PPT. See, this one is the role of fungal bead biodegradation of emerging pollutes. This one advance in chemical, medical, cosmetic, plastic productives industry have improved in agricultural yield, health and human life in general. As a negative consequence, chemical are internally and unitarily released to the terrestrial aquatic environment. Sometimes effect for the entire ecosystem. Okay. In that one, sometimes to effect for the total entire ecosystem, entire the ecosystems. Now, one migrate strategy to counteract this pollution is bioremediation. Bioremediation is the biological mediation process during undesired compound. This is a transform, degrade, sequestrate, and entire remove the from the ecosystem. That's why organism across all domain of the life biodegradation. The fungi, particularly promising candidate, the process metabolism, break down complex molecule, the make fungi ultimate degradation of the reactions organic matter. This is the just introduction. This one. What is the emerging pollutants in the large scale manufacturing using application of pesticides, pharmaceutical, personal care products, plastic polymers, and heavy metals. Heavy metals. These compounds are designed to safety ever-growing need for a better life, health, and ways to ensure food and material supply for a growing human population. However, their however, their production and application are often accompanied by the waste generation. This is not parallel by migrate stages. This is the through transport in groundwater, rivers, air, ocean. These are recent reclient products compound have reached and contaminated virtually all ecosystem, that is earth. For example, organic pesticides have been detected in deep sea sediment of the Pacific Ocean. Pharmaceutical and personal care products also frequently dedicated in wastewater treatment system as well as marine ecosystem. Microplastic have been found in remote places such as Sea of Antarctica and deepest part of the marine realm. marine trend and nanoplastics also are seemingly widespread to that one. In nature, these emerging pollutants causing adverse effect ranging from impact on the organism to the ecosystem level. For example, include bioaccumulation of methyl mercury in shellfish, causing poisoning in human upon consumptions. In ingestion and marine organism in plastic litter, increase in antimicrobial resistance gene due to the excess use of antibiotics and their indicate disposal, and finally deserve ecological consequence due to the large scale use of pesticides, dichloro-diphenyxy, trichloroethane, DDT, that is called the DDT, dichloro-diphenyl trichloroethane. Before it is banned in many countries. In order to migrate the increasing amount of and affect these contaminants. Biological process can be utilized to break down, transform, remove hazards, pollutant from the environment. These processes have been coined bioremediations. This is the term is called the bioremediations. These bioremediations first applied George Robinson in 1960 by using microbes to migrate the effect of soil split. Bioremediation mostly relies on plants, that is a phytoremediation, or microorganism, that is a microremediation, to degrade pollutants. This prominent example for bioremediation in aquatic environment are the use of microalgae that accumulate heavy metals, pharmaceuticals. Microbioremediation effort benefits from the large natural diversity of microorganism, features a spectrum of pathway to metabolism, co-metabolites, wide range compounds. These are the bioremediations, how to control. In fungal bioremediation agents, what are the fungal bioremediation? You have studied the fungal, no? In the fungal bioremediation, what type of fungal bioremediations, what type of organisms are increased in this one? You can study this one. Here, main important of this one, fungi are organisms gaining increase attention because of their increase attention because of their broad proteins and broad protein potential for bioremediation application. Fungi are diverse kingdom in the tree of life belonging to domain eukaryotics, nevertheless fungal taxonomy. This is the most recent taxonomical update. 19 major phyla involved in the fungal biotechnology. That is a bioremediation method. One is a Aphidomycota. Second one, Ascomycota. Third one, Basidiomycota. Fourth one, Basidiomycota. Basidiobolo-mycota. Basidiomycota. Blastocladiomycota. Next one, sixth one, Carisporoliomycota. Sixth, fifth, seventh one, Trichomycota. Next one, Critidiomycota. Next one, Belmorphomycota. Next one, Mortirellomycota. Next one, Mucoromycota. Next, Neocali mastigomycota. Next one, Ophidiomycota. Next one, Rosellomycota and Zoophagomycota. These are 19 major phyla of fungi involvement in the biodegradation agents. The number described species 1 million, but total number of fungal species is estimated to range between 2 to 4 million fungi, ubiquitous heterotrophic organism prevalent throughout terrestrial and aquatic environment. Nevertheless, fungi have been studied in terrestrial environment more extensively. This one is the uh, fungal degradation of pest. How to control the pest? Increased global food demand and the need to provide global crop losses due to the pest have resulted in the extensive annual utilization of about 2 million tons of pesticide worldwide, not in the our country. Total worldwide, the pesticides to production 2 million tons. Pesticides are deliberately applied to kill unwanted plants. That plants is called weed plant. Insect control, rodents, and other living organism threatening the cultured crops, many cultured crops. However, it is postulated that approximately 90% of agricultural pesticide do not reach the intended target organism, but instead disappear the environment. Indeed, pesticides have been dedicated in the atmosphere, soil system, groundwater, and ocean as a result of high population density and intensive modern farming practices. The pesticides such as organochlorines, organophosphates, pyrethroids, carbonates are associated with detrimental effects including high environmental persistence, bioaccumulation, long-range transmission, and adverse effect to the non-target organism. Exposure to pesticide can, for example, cause different types of human cancer, tetrogenic and genotoxic defects as well as endosarin and never diffusion. Pesticide may be cause acute lethal effects also. These are the harmful for the pesticide using the fungi. See, this is the schematic overview. Mycoremediation in the environment, pollutant, commonly used fungal enzyme. Fungal enzyme classes are highlighted, pollutant, follow intracellular and extracellular enzymatic degradation, leading to the production of diversity of metabolism and degradation. Coming to the fungal degradation in the pharmaceutical personal care products. Pharmaceutical and personal care products, PPCPR, an emerging class of contaminants as modern societies are increasingly focused on health, hygiene, and personal care. A substitutional fraction of PPC ends up the nature, for example, via sewage streams and wastewater treatment system. In fact, there is a lack of regulation regarding use and discharge of many PPC. This arise concern in particular related to potential adverse effects on ecosystem, and human health in if pharmaceuticals involved. The pharmaceutical compress waste number of substance are there. These are the fungal have been break down the range of pharmaceutical ranging from antibiotics, anti-inflammatory drug, anti-cancer drug, anti-depression, diuretic, allergies, and beta blocker, the most study fungal group able to degrade pharmaceutical or WRF. For the degradation of pharmaceutical, these fungi make often use the same intracellular and extracellular enzyme that they also use for the degradation of pesticides. This is the fungal pesticides to how to control the fungal pesticide. Next one, conversion in plastic polymer degradation by fungi. Conversion in the plastic or synthetic polymers. All plastic are the synthetic polymers, commonly of petrochemical origin. Petrochemical origin, large scale production of plastic begin in the 1950s and exponentially increased until today. Plastic are used in nearly all industry sectors, most important packing, textile, electronics, and consumer production. Plastic polymers production 2018 reached 359 meter MT. However, waste mismanaged and country specific inadequate policies lead to the disposable plastics in landfill or direct littering in nature. Of the global plastic production, it is estimated that a 1.8 to 4.1% entered the ocean via coastal deposition of riverine input. Through the atmosphere deposition, plastic litter in the ocean has raised concern both the scientific and public domains and was identified as an environment threat. In the ocean, ocean plastic may be float at the surface or sink the seafloor depending on the polymer density and shape indeed. It has been suggested that sedimented plastic debris might become a stratigraphic indicator of Anthropocene epoch. Some plastics are ingested by marine also. Nevertheless, the ultimate fate of marine plastic is still large unknown. Global calculations of floating marine plastic debris only account for a few percent of expected amount that has entered the ocean. Several reasons for the this missing plastic paradox have been discussed. Most important, stress and weathering lead to the fragmentation of large plastic items into even smaller plastic items. This is very plastic to control the effect of the total environment. Assessment of fungal diversity depend the environmental and experimental factors. For example, salinity is a significant factor. Salinity is the main it can observe to the way in the ocean countries. One second. Sorry for the disturbance. My voice is not assessment of fungal diversity. The may depend on a variety of environmental and experimental factors. For example, salinity is a significant factor influencing fungal communities, as shown in the Baltic Sea. Baltic Sea. Among experimental factors, the selection of primers, sequencing depth, and reference data bases will strongly influence phylogenetic resolutions. The potential influence of using different molecular marker genes to analyze fungal communities on plastic was addressed by comparing three genes are there. ITS2 one gene, 18 srRNA, B4 is another one, and 185 rRNA V9 another one. These were used for analyzing the fungal community on floating plastics from the western South Atlantic and the Antarctic Peninsula. The study found that by using 18S V4 primers, only 2.2% and 4.3% reads could be assigned. By using primer targeting the 18 years, a similar low percentage was assigned in stark contrast primer. This is the worked much more allow assign 60% Atlantic Ocean, 80% Antarctica Ocean. In total, 64 fungal orders were associated with plastic. The fungal community of the different plastic was highly diverse and no difference were found between polymer types, shape, size, size, classes, or even sampling station and location. However, novel fungal phyla, Aphidomycota, Juphagomycota, Mucoromycota, and Blastidocalcidomycota were reported for the first time as part of the biofilms on marine plastics. This is the very important and useful, as the fungi are the mainly an important part of the microbial biofilms coating plastic fragments in the marine realm. It is this one is a fungal burner. Next one, fungal biodegradation of bio-based biodegradation plastics. This one, biodegradation plastics considered to as the mostly industrial process. This one you can observe, this one is a mostly industrial process. In the industrial process, how to, this is the control, and yeah, this one is which is the real temp. However, this mostly include industrial process, evented temperature or moist content, which often do not resemble conditions in the nature. Okay, this does the even when certain plastic type certified biodegraded plastic, it might be only degraded plastics. Bio-based raw material for bioplastic productions originating from renewable source, vegetable oils, cornstarch, soya bean, protein, sugar, potatoes, even microbes. This common misconceptions is that bio-based plastics are by default biodegradable, whereas fact also conventional recalcitrant plastics we use this one. This is a total and micro remediation of heavy metals. Heavy metals are generally malleable or ductical elements, but no clear definition exists that well defined the term heavy metals. Heavy metals may be described as dense material forming a block of almost all elements in groups 3 to 16 that are in period 4 and greater of the periodic table of element. The block of earth and rare earth elements as well as titanium, aluminium, silicon may then be described as the light metals. Almost all metals are scarce. All metals are scarce and unevenly scattered in the earth crust, but may be concentrated in certain region as a result of a geological anthropogenic process. They are also transported by natural phenomena or anthropogenic activities. For example, arsenic, nickel, human hair. These are atmosphere, aquatic environment. Most heavy metal process risk almost any ecosystem on earth and human health problems also. For example, what are the heavy metals? Are they arsenic, nickel, thallium, cadmium, lead, mercury are notably harmful or poisonous. Potential pathway, heavy metal contamination, exposure are many flood but are often related to erosion of geogenic source and mining for metals or combustion of fossil fuels, disposable of industrial waste. High level of heavy metals are also found in certain agricultural products and paints from which they leach into the environment. Heavy metal generally persist in nature of for long periods of time and may be accumulate in plants, animals such as arsenic in rice and mercury in fish. They are also subjected to trophic transfer and eventually pose a considerable threat to human health. Mm. Heavy metals toxicity has been characterized for the plants, animals, human bodies, microbes also, where the metal can causing the damage DNA, denatural proteins, inhibition of the enzyme activity, inhibition of the cell division, and disturb the cellular membrane. That's why nowadays the increase the cancer cases also. Why? These are the effect for the human body and animals also. Okay. Heavy metals in contrast to organic pollutants cannot be degraded, but their oxidation state may be changed, which can change their toxicity and mobilize or precipitable them. Thus, bioremediation of heavy metals release on bacteria, microalgae, fungi tolerant to heavy metals. Among these organisms that are able to assimilate heavy metals and thereby elevate the pollution in nature or preferably choose bioremediation applications. Fungi process extra and intracellular biochemical and molecular mechanisms which relay on firstly binding the metabolism. Firstly, binding the firstly binding the metabolisms. This is the mostly important for the fungal that one. And the next one is the one second. Next one and compulsory component. Next, extracellular mechanism involvement is there. Intracellular mechanisms also present in that one. Extra mechanical mechanism instead to particular that is the entrance to toxic metals. This one is the binding or biosporation considered one of the first extracellular barrier. Here, metal toxicity in fungi. For instance, proportion of CD binding cell wall packs that is called the Bacillus involvatus was similar to that found intracellularly. Extracellular release of supposed enzyme or pollutant cheating agents. What is the intracellular mechanism? That is called the Bacillus involvatus was similar to that found here. Main important of this one, intracellular mechanism. Yeah, this one is the total bioremediation of the fungi. Application potential of the fungi. Here, various stages have been suggested to bioremediation, pollutant in soil, sediment, and aquatic environmental. Soil one, sedimentation method, aquatic. The application microremediation can be conducted on soil, referred as the microremediation by the removing of the contaminated soil, sediment, water matrix to a different location where the microremediation in carried out ex situ. Most are the sometimes in common in situ also. In situ bioremediation methods are bioaugmentation, bio-overting, biosparing, and natural alternations. Ex situ and in situ, both methods in the here nature and extensive of the pollution association for available suitable conditions. See, this is the picture. You can observe that one, that picture. This is a illustration of most commonly applied in situ and ex situ microremediations strategies. Biovoting and biospring addition of oxygen to promote activity of aero of activity of activity of aeromicrobes, biostimulant addition to nutrient that promote microbes remaining the pollutant, addition of microbes to site the ex situ stages. This is a total ex situ and in situ bioremediations useful by fungi. Next one, mycorrhiza based on enzyme experiences and immobilization. Ascomycotina. First one useful the fungi is the which one? Ascomycotina is the most important fungi. Ascomycotina, penicillium, yeast, all are the industrial fungi and very useful fungi. The Ascomycotina fungi are candidates for genetic manipulations and gene expression of enzyme for the upscaling of remediation capability. Some candidate enzyme are lacases and triosinase. Lactoses కాదు ఇది lacases. Lacases, triosinases with a large range of application, peroxidase from basid and this is the ascomycetes. Another one, basidiomycetes have already been used for commercial application with enhanced resistance to peridoxide and a wider redox septum for degradation through modification. Recent study have been focused on this process of the bioremediation process. The conclusion for future perspectives, these are the total bioremediation. Another one I'll explain to the environmental monitoring also present in the fungal. How to environmental for control that one. These are things also many environmental is to useful this fungi contaminate and cultures used for the environmental methods also. Today we will discuss about to the total class for the fungal, environmental, fungal, biotechnology and quality. All are the things are completed in this class. Main environmental fungi also, the valuable introductions, all are the complete that one. Main atmospheric fungi, both outdoor, indoor, atmospheric, outdoor and indoor atmospheric. That is the species. What type of species in fungi? Cladosporium, penicillium, Cladosporium, penicillium, Aspergillus, and Alternaria. By east, Mycelia. Cladosporium is always the dominant fungi in outdoor. Okay, atmosphere and indoor atmosphere of normal and healthy building, except hospital where aspergillus fungi, sorry, penicillium are useful dominant. The abundance of the fungi, other fungi, varies with the season and place. In relation to outdoor environment, indoor atmosphere typically uh, display lower diversity and abundance of fungi. The following genera can be represented indoor but are always clear minority. Apicida, Acrimonium, Arthenium, Erubaceum, Viveria, Botrytis, Candida, Ketomina, Chrysosporium, Epicoccum, Epicocum fusaria, Gleocladinum, Mucar, Nigrospora, Passixlomyces, Phoma, Rhizopus, Scopulariopsis, Sporobolomis, Symphalus, Trichoderma, Euloderma, Vericillium. These are the example for the useful. Okay, how to climate and human activities are the main factors that influence the composition of outdoor atmosphere. In the temperate climate, these display a typical pattern around the year on the country. Climate is not determinative in the microflora indoor atmosphere, but human activities and the quality maintenance of the building to play major role these environment for this reason. Dominant indoor vary between building can be used the atmospheric fungi. Here, outdoor, indoor roots also different process in the fungal. How to fungal growth, sporonation, adaptation. How to growth, adaptation, xerophytic condition. Fungi from atmosphere, indoor environment, influence temperature, humidity, atmosphere relative humidity, and substrate moisture content also. The optimum temperature for growth and sporulation is usually around the temperature minimum 25 to 30 degrees centigrade. 25 to 30 degrees centigrade. Low higher temperature result in lower growth and sporulation rate. A remarkable concept, expectation compression fungi. These are the use for the total fungal fragment and sometime allergetic tools also. See, this is the total fungal classes, fungal biotechnology. Today class total complete the biotechnology. Okay. If any doubt, if you call me or message me, I'll clarify your doubts in the total fungi. 2 units are completed. Next class, we will discuss about to the plant pathology method. How to plant pathology use. These are things we will discuss about to the next class. Okay. Nawala ka mo.